Welcome to this explainer. If you've ever felt like there is this massive gap between reading a dense biology textbook and actually scoring full marks on an exam paper, you are in exactly the right place. Today, we're going to bridge that gap by decoding a real trial paper. Specifically, we're taking a close look at question 2 from the CADA biology trial paper. We're going to break it down, build up our fundamental knowledge, and make sure we secure every single mark.
Here is our roadmap for today. First, the CADA trial question. Second, water and essential elements. Third, building biological polymers. Fourth, genetics and DNA structure. And finally, solving the exam question.
Section 1, the CADA trial question. Let's establish exactly what we're trying to solve today.
Take a close look at the structure we have right here. We can clearly see a double helix structure, which immediately tells us we're dealing with a specific type of nucleic acid. But pay special attention to those parts labeled P and Q. The exam actually asks you to name these two exact components.
So, the big question is, how do we correctly identify parts P and Q? And in the second part of the question, what is the biological effect if the transcription process just fails? To answer this accurately and with absolute confidence, we need to pause the exam for a second and dive into the essential fundamentals of Chapter 4.
Section 2, water and essential elements. Let's see how this builds our foundational understanding of biological molecules.
Well, before we can build complex nucleic acids, we really have to talk about the medium where all these biochemical reactions happen, right? Water. Water molecules are polar, meaning they have a slightly negative oxygen end and slightly positive hydrogen ends. This polarity allows them to form hydrogen bonds. Because of these bonds, water acts as a universal solvent. This is absolutely critical for transporting solutes, like glucose, across plasma membranes. Plus, its high specific heat capacity of 4.2 kilojoules per kilogram per degree Celsius helps organisms maintain a stable body temperature, while cohesive and adhesive forces allow for things like capillary action in plants. Within this watery environment, cells build organic macromolecules. So the crucial point here is memorizing this exact elemental breakdown. Carbs and lipids? They're made of carbon, hydrogen, and oxygen. Proteins step it up by adding nitrogen, and most even contain sulfur and phosphorus. But look at the bottom row, nucleic acids. They consist of carbon, hydrogen, oxygen, nitrogen, and phosphorus. You absolutely need to know this makeup because nucleic acids are exactly what our trial question is testing.
Section 3, building biological polymers. Now that we know the elements, how do cells actually build these complex structures from scratch? Cells use two very specific textbook processes. To build a polymer, they use a process called condensation. This is where monomers are linked together by removing a water molecule. Conversely, when the body needs to break these complex polymers back down, say during digestion, it uses hydrolysis. Hydrolysis literally means splitting with water. It adds a water molecule right back in to break those bonds. This illustrates beautifully how these processes apply across different macromolecules. Simple monosaccharides like glucose undergo condensation to become polysaccharides like starch or glycogen. Amino acids link up to become polypeptides. And one glycerol molecule combines with 3 fatty acids to form a triglyceride, or fat. Recognizing how these monomers snap together is really the key to understanding the molecular machinery of life.
Section 4, genetics and DNA structure. Let's zoom in on the specific class of molecules needed to solve our exam question. Just like glucose is the monomer for carbohydrates, the nucleotide is the monomer for nucleic acids. Every single genetic code starts with this basic building block. A single nucleotide always consists of 3 parts combined through condensation: a 5-carbon pentose sugar, a nitrogenous base, and a phosphate group. Now, there are 2 main types of nucleic acids, and you have to know the difference. On one side, we have DNA, or deoxyribonucleic acid. It features that famous double helix structure, a deoxyribose sugar, and its nitrogenous bases include adenine, guanine, cytosine, and thymine. On the other side is RNA. Notice the strict structural differences here? RNA is a much shorter, single polynucleotide chain. It uses a regular ribose sugar, and most importantly, it swaps out thymine for uracil. And how does this massive DNA double helix fit inside a microscopic cell nucleus? It's honestly amazing. The DNA polynucleotide chains wrap tightly around specialized proteins called histones.
This brilliant DNA histone combo forms structures known as nucleosomes. Then, those nucleosomes intertwine and coil incredibly tightly to form the full chromosome structure, elegantly packing our entire hereditary blueprint right into the nucleus.
Section 5, solving the exam question. Okay, we have our toolkit, so let's return to the trial paper and secure those full marks. Looking back at the problem, we see two polynucleotide chains intertwined in a double helix. This immediately confirms we are looking at DNA. Now, let's identify P and Q. P is pointing to the weak bonds connecting the two rectangular bases across the middle. Q is pointing to the pentagon-shaped structure forming the backbone alongside the phosphate group. Using our textbook knowledge, we can totally nail the official marking criteria. P represents the hydrogen bonds. Remember how we talked about water forming hydrogen bonds? Those exact same types of bonds connect the complementary nitrogenous bases, adenine to thymine and cytosine to guanine, holding the two DNA strands together. Q is the 5-carbon sugar. Since this is a double helix, it's DNA, meaning the exact answer for Q is a deoxyribose sugar, though the marking scheme actually accepts the general term pentose sugar too. Boom, 2 marks secured.
Now let's tackle the second part of the question. We're looking at protein synthesis here. DNA undergoes transcription to form mRNA, which is then translated to form a polypeptide chain made of amino acids like methionine, arginine, and tyrosine. The exam question asks, explain the effect on protein synthesis if the transcription process fails to occur. The textbook terminology gives us a very clear logical cascade. According to the marking scheme, if transcription fails, your first point is that mRNA simply cannot be formed, meaning there are no codons. Point 2, without mRNA, the translation process cannot occur at the ribosome. And point 3, the ultimate result is that there's no protein synthesis, meaning 0 polypeptide chains are produced. By clearly stating this chain reaction, you secure the remaining marks perfectly. We've easily solved the exam question by relying purely on our structured textbook knowledge. But before we wrap up this explainer, it's pretty fascinating to think about the bigger picture, isn't it? If a single missing hydrogen bond or one tiny error in the transcription process can completely stop an organism from producing vital proteins, how does life maintain such incredible precision billions of times a day? It really leaves us to marvel at the immense, microscopic machinery driving our biology. Keep that in mind as you continue studying. You aren't just memorizing facts, you are decoding the very blueprint of life.